Good afternoon. My name is Laura Sofia Vera Leon, a chemical engineering student, and the word to present is fermentation and its types.
Good afternoon, my name is Nicolas Guerrero. Our presentation is about metabolic pathway of fermentation.
In general terms, when we talk about fermentation or fermentative metabolism, we are talking about a catabolic process in anaerobic conditions, which means that it is a process where molecules are degraded, usually carbohydrate or sugar, in absence of oxygen. Then within the fermentation we find four main ways: lactic, alcoholic, butyric, and acetic. Fermentation the best known for the wheat used in industry are the lactic and alcoholic routes. The lactic fermentation pathway produced mainly by bacteria of the Lactobacillus genus that convert carbohydrate such and glucose and other exhaust into lactic acid. Alcoholic fermentation is caused by the activity of some microorganisms, generally yeast, that process carbohydrate such and glucose, fructose, or sucrose to obtain ethanol and carbon dioxide as final products. The butyric fermentation pathway is the conversion of carbohydrates into butyric acid by the action of bacteria of the genus Clostridium. It is normally produced from lactose with the formation of butyric acid and gas. And finally, before going on to detail the main pathway, we have bacterial fermentation by Acetobacter, which is a genus of aerobic bacteria that convert ethyl alcohol to acetic acid.
Microorganisms involved. Lactic fermentation. Lactic fermentation is caused by some fungi and bacteria. The most important lactic acid that bacteria produce is Lactobacillus. Other bacteria produced big lactic acid are Leunostococ, Mesenteroides, Pediococcus, cerevisiae, Streptococcus, lactis, and Bifidobacterium bifidus. The presence of lactic acid produced during lactic fermentation is responsible for the wider tasted and for improving the stability and microbiological safety of the food. Microorganisms involved. Alcoholic fermentation. Alcoholic fermentation requires microorganisms to transform sugars into ethanol and carbon dioxide. Although the most commonly used microorganism is the yeast Saccharomyces cerevisiae, there are other microorganisms that can carry out this type of fermentation. Two microorganisms, Saccharomyces cerevisiae and Simomonas. Saccharomyces cerevisiae yeast has enjoyed a long and distinguished history in the fermentation industry due to its efficiency in producing ethanol. Chemical reactions, lactic fermentation. Number 1. Lactose is hydrolyzed by the action of lactase, giving glucose and galactose. Galactose, in turn, is isomerized, giving glucose. Number 2. Glucose by glycolysis is transformed into two molecules of pyruvic acid, also releasing 2 ATP and 2 NADH more H. Number 3. Pyruvic acid, which is the last electron acceptor, is reduced by the action of NADH more H obtained in glycolysis and transformed into lactic acid. This reaction is catalyzed by lactate dehydrogenase. Chemical reaction, alcoholic fermentation. Number 1. Glucose by glycolysis is transformed into two molecules of pyruvic acid releasing 2 ATP and 2 NADH more H. Number 2. Pyruvic acid undergoes decarboxylation, loses CO2 and is transformed into acetaldehyde. This reaction is catalyzed by the enzyme pyruvate decarboxylase, which is not found in animal tissues. Number 3. The acetaldehyde that is the last acceptor of E and H more is reduced by the action of NADH more H that was obtained in glycolysis and is transformed into ethanol. The enzyme is alcohol dehydrogenase. Number 4. The final balance of alcoholic fermentation is glucose more 2P more 2 ADP and 2 ethanol more 2 CO2 more ATP more 2H2O.